right, now that we know a little bit of the vocabulary that we're going to be using, and you know about the different types of cells in your body, let's get into the step-by-step of the cell cycle. Let's break it down. Let's start by looking at the big picture. What does the whole process look like? Okay? Don't worry about drawing this right now, because it'll make more sense if we go through it step by step. I just wanted to give you a preview, and then you can draw these as we go step by step. That way the picture will be next to the notes. Here's what these guys look like in a real cell. These are real cells that we've taken pictures of. Okay? Again, this will make more sense once we've studied it, so you can always come back to this. All right, so the cell cycle, the whole process, basically just the cell living its life is the cell cycle. It involves the cell growing and developing and then dividing. The cell cycle has 4 main stages. The cell's going to go through G1, S, G2, and M. That's the big overall picture, okay? The whole process that the cell is going to go into. Now, some cells will enter a special phase called G0 or G0. And what that means is that the cell's just never going to divide again. These are like your brain cells. Once they go through the process a few times, they're going to go into a special thing called G0, which means they're never going to divide or again. They're just going to live their lives and do their jobs. But most cells are going to go through the cell cycle, G1, S, G2, and M, and then start all over again with G1, S, G2, and M, and so on, and so on. So the first part of the cell cycle is called interphase. This is the longest phase of the cell cycle. The cell spends about 90% of its time in interphase. Okay? Now, I just said that there was 4 parts of the cell cycle. 3 of those 4 parts belong to interphase. That's how long it is. Okay? The G1, the S, and the G2 of the cell cycle are all part of interphase. Okay, so let's talk about what's going to happen in each one of these smaller parts. So G is going to stand for growth. Okay, G1 equals growth. The cell is going to grow, and it's going to carry out its normal functions. So if it's a liver cell, it's going to grow and help filter your blood. If it's a heart cell, it's going to grow and help pump the heart and get the blood through the system. If it's a stomach cell, it's going to grow and help produce digestive juices, etc., etc. We're also going to have our first checkpoint. Remember, we have to regulate cell growth. We have to regulate the cell cycle. So we're going to have a checkpoint in G1 for the cell to go, okay, am I healthy enough to think about dividing? Am I big enough to think about dividing? And this first checkpoint is known as the point of no return. If the cell decides to move on with cell division, it decides it's healthy enough and big enough to divide, and it starts the process, it either has to finish the process or it must die. There's a special sciencey word for when a cell makes the decision to die, and that is apoptosis. Apoptosis means programmed cell death. This is where the cell willingly kills itself to prevent something horrible from happening. So if it detects that it might have something terribly wrong with it and become a cancer cell, it will go through apoptosis and kill itself. Sometimes cells, if they're infected with viruses and they're able to, they'll go through apoptosis so that they're not taken over by viruses and reproduce the virus. Okay? So make sure you know that word, apoptosis, programmed cell death. Okay? So this checkpoint is the point of no return. The cell will either finish the process of division or it will go through apoptosis and die. There's no going backwards from here. So after G1, we're going to enter S phase. S stands for synthesis. What are we going to synthesize? Remember, synthesis means to put together. Well, we're going to synthesize DNA. We're going to do DNA synthesis, also known as DNA replication. Now, we've already studied the process of replication where you untwist, unzip using helicase, fill in with missing nucleotides to create 2 full strands of DNA, and then rezip and retwist. If you don't remember that, you need to go back and rewatch DNA replication videos, or you need to come to ILT for help. We're at the point in the semester where we're starting to bring a lot of things together, and we're seeing how things are building on other things. Okay? So, part 2 of Interphase is synthesis, where we're going to do DNA replication. Now we're going to have 2 copies of the DNA. We're going to have another checkpoint here, where we're going to proofread the DNA. Did we copy the DNA correctly, or did we make mistakes? Do we need to fix mistakes? Okay? So at this point, we could pause so that we can fix mistakes, or we could say, oh, too many mistakes, time to die. Or everything is right, or everything was fixed and is now right, and we can move on to G2. Again, G for growth. G2, we're going to grow some more, or make sure everything's ready to go. We're going to prepare to move into M phase. And for now, M is going to stand for mitosis. Mitosis. Remember, mitosis happens in my toes. So these are your body cells that are going to be going through mitosis. Here's our final checkpoint for the cell. Am I big enough to divide? Is my DNA ready to go? Am I healthy? If the answer is yes, you move on. If it's no, but it's something small that we can prepare for and fix, then we might pause here for a moment to get ready to move on. Or if it's something catastrophic, the cell will die. So here's what interphase looks like. During interphase, the cell's DNA is all loosey-goosey. Okay? It's in the form of chromatin. Okay? We're going to focus here on the nucleus. Okay? So we have our nucleus. Notice we have a full nucleus. The nucleus is in place, and inside it is chromatin. The DNA is loose and looks like a bowl of spaghetti. You need to be able to recognize the phases from pictures. You need to be able to put the pictures in the correct order. So make sure that you are studying to prepare for those things, and we'll do some activities to give us practice. All right, let's move on from interphase into M phase. The first phase of M phase is prophase. Remember, pro means first. So this is the first phase of mitosis, prophase. So what's going to happen? Because you need to be able to tell me what's happening in the pictures, what's happening in each phase. So in prophase, our chromatin, the bowl of spaghetti DNA, is going to condense. It's going to tighten up, and it's going to turn into chromosomes, the X shape that DNA can take on. Okay, so if you look at the cell now, it's going to look a little bit different. Okay, it's kind of going to look like worms now. It's not as loose. It's tightened up. Okay, and in drawings, they'll actually draw X's for you so you can recognize it. Our nucleus is going to start to disappear. That's why here in the drawing you see it represented by a polka dotted line to represent that it is disappearing, it is going away. Our centrioles, they're now going to do their part. Remember when we studied centrioles previously, we said they just kind of hang out in the cell most of the time until the cell's ready to divide. Now the centrioles are going to start migrating. They're normally hanging out near the nucleus. Now they're going to go to opposite sides of the cell. Remember, centrioles are only found in animal cells. And we're going to start making spindle fibers from the centriole. Spindle fibers are long proteins, and they're going to be used to rip the cell apart. Okay? So make sure that you know that all of these things are happening during prophase. After prophase comes metaphase. Meta for middle. The main thing that's going to happen here is that your chromosomes are going to line up in the middle of the cell. So metaphase, middle. Also notice that your nucleus is totally gone. No more nucleus now. Okay, the nucleus is just going to get in the way, so we got to get rid of it so that we can divide up all this DNA. Okay, so notice chromosomes are in the middle, still X-shaped. If you look at a real cell, the DNA is wiggly, so you can't see the X shape, but you can still see the dark line where they're lined up in the middle. Your spindle fibers are going to attach to the centromere. Remember, the centromere is the center of the chromosome. Okay, it's where the two sister chromatids are attached. So the spindle fibers are going to attach to this point so that we can rip them apart. Metaphase is followed by anaphase. During anaphase, your chromosomes are going to be ripped apart. So now your chromosomes are turned into sister chromatids. Okay, they are ripped apart. The way I like to remember this is when they're getting pulled like this kind of looks like the letter A if you put a bar right here. Okay, so anaphase starts with A, and the chromosomes look like you're starting to write the letter A. That helps me. Okay, or you can say they look like creepy Freddy Krueger fingers reaching towards one another. Whatever works for you. Okay, so anaphase, the chromosomes get ripped apart into sister chromatids. Remember, all these vocabulary words I'm using we studied last unit, so make sure that you're studying them if you're struggling, or come to ILT for help. Okay. Next, we have telophase. Telophase, we're getting near the end. Okay, we still have our chromatids condensed like little wormies, but our nucleus is starting to come back. Notice the polka dotted line. Okay, the nucleus is coming back, but now we're going to have two of them. Okay, because we're starting to make two cells. Spindle fibers are gone. They have broken apart into the cell. And your centrioles pair back up. They go back to looking like they're doing nothing for a while. Okay, and the cell is starting to pull apart. So during telophase, it's really easy to recognize because it kind of looks like a peanut, or I say it kind of looks like a butt during telophase. Okay, and our very last phase, and there's a little bit of argument whether this is a true phase or not, is cytokinesis. Cytokinesis is super fast, and it is just where the cell is literally dividing. It's dividing its cytoplasm and turning into two cells. This is super fast, like snap your fingers, split second. So some argue that it's not an actual phase, and some say it is a phase. So just be aware that sometimes it is, sometimes it isn't, depending on who you ask. Okay, so cytokinesis is the actual just breaking it up, and now you have two identical daughter cells. They are identical to the original, they are identical to each other, and they start immediately all over in interphase G1. Okay? Here, animal cells and plant cells are going to be a little bit different. Animal cells are going to create a cleavage furrow because they're soft and squishy. So it pinches in, starts to look like a butt or a peanut, and just goes all the way through until it divides. Cleavage furrow. Plant cells, because they're rigid, they have a cell wall. They're going to create what's called a cell plate that divides the two. Okay, so a cell plate starts to form until it turns into the cell wall. Okay, let's look at some pictures of this in real cells. Okay, so here's your cleavage furrow. It's soft and squishy, so it's squeezing in. And it's just going to keep on squeezing all the way through until it pinches into two cells. Okay, plant cell, solid and rigid. So you see this thin line right here, that's the cell plate. And it's just going to keep growing until it turns into a cell wall. Okay, so there's a difference between your plant and animal cells. Animal cells have centrioles, plants don't. Animal cells use a cleavage furrow during cytokinesis. Plant cells use a cell plate during cytokinesis. Make sure you can tell those differences. And here's your list. Okay. Finally, some people like to have these little memory mnemonics to help them remember. So here's some sentences to help you remember the correct order of the cell cycle. I like this one. Impatient people may attack teachers constantly. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Okay? Important people must analyze tasks correctly, or you can make up your own sentence if you don't like these ones. But make sure you know the correct order of the phases of mitosis. Make sure you can put pictures in the correct order. Make sure you can describe what's happening. There's a lot we need to know, but we're going to get more practice with it.